Hi, this is Dr. Neela Bakore, and in the previous segment we talked about the dark reaction, that is Kelvin cycle, and we said that the one which we were discussing was the easier option or the easiest version. In the sense, first we need to understand what exactly is going to happen, and now what we are taking is the main steps which take place during Kelvin cycle. But to have a comparison in front of us, we will quickly draw the simple version of Kelvin cycle, and then we will take up the complicated one in which all other intermediate steps we would discuss. So, topic is our photosynthesis. And we are talking of Kelvin cycle. The easier version or the simplest version which we already discussed was, we said that carbon dioxide is received by RUBP. This first step is called carboxylation. The enzyme here is Rubisco, which is the most abundant enzyme or protein on the earth or globe. And then we said the first thing or compound which is formed is a temporary 6 carbon compound. Let us skip that because we just want to have a comparison when we draw the complicated Kelvin cycle. So here 3 carbon compound, that is phosphoglyceric acid, is formed. This phosphoglyceric acid then changes into diphosphoglyceric acid, and the phosphorus, that it comes on 2 carbons, that is carbon number 1 and carbon number 3. Then this phosphoglyceric acid, diphosphoglyceric acid, undergoes reduction with the help of NADPH2, the reducing power which is synthesized during light reaction. This NADPH2 gets oxidized into NADP, and this phosphoglyceric acid undergoes reduction. It gets reduced into phosphoglyceraldehyde, PGAL. This molecule was a diphosphate, and here phosphoglyceraldehyde has only one phosphorus. So, in this process inorganic phosphates are also given out. This PGAL, it bifurcates into 2 parts. One molecule or a part of it will form glucose, and the remaining PGAL will help in regeneration of RUBP. Again, PGAL is with single phosphorus and RUBP is with 2 phosphates. So, here also phosphorylation takes place. So, ATP gives that phosphate an energy and ADP is given out. Now, let us put the numbers which we started with. We said 6 CO2 and 6 RUBP will accept this CO2, 12 of these. So, everywhere everything was coming to 12. Here 2 PGAL, let us write 2 PGAL separate, which give us 1 glucose, and these are the 10. So, out of 12 which are formed, 2 separate out of the cycle and 10 help in regeneration of RUBP. So, this is what was the simple version. Now, we are talking of the multiple intermediate steps which actually take place in Kelvin cycle. We will draw certain things which are common in both. So, the common thing is that carbon dioxide is accepted by same RUBP, that is rubulose biphosphate, enzyme that helps us same Rubisco. Here also the first compound which is formed is a temporary short-lived 6 carbon compound. So, a 6 carbon compound, it is temporary and short-lived. That means as soon as it is formed, it is going to dissociate. This 6-carbon compound changes into a 3-carbon compound, that is phosphoglyceric acid. This phosphoglyceric acid, we are writing the steps which are common. Phosphoglyceric acid changes into 1,3-diphosphoglyceric acid. This undergoes reduction to form phosphoglyceraldehyde, and here that ATP is being used because from phospho, that is single phosphorus containing molecule, 2 phosphorus containing molecule is formed, and in this case reduction is taking place. So NADPH2 changes into NADP and acid gets reduced to aldehyde. So far is same as what we did in the simple version of Kelvin cycle. Now let us talk about this RUBP regeneration. Rubulose biphosphate, it is formed from monophosphate. So it is called rubulose phosphate. So rubulose phosphate gives rise to biphosphate. Rubulose phosphate, single phosphate containing molecule, and biphosphate has 2 phosphates. So here this ATP and all those things we will add little later. According to all this which we talked of in the simple version, how many of these molecules are formed? 12 molecules of diphosphate, 12 molecules of PGA. So we are taking the same number, 12, PGAL. Now all these aldehyde molecules or phosphoglyceraldehyde molecules, they do not participate in a single pathway. Here what we showed, that all of them undergo phosphoglyceraldehyde. They divide into 2 parts, one gives glucose, the other helps in regeneration. They separate into 4 different groups, or groups in the sense now they will participate in different pathways. 2 molecules will participate in a different manner, 5 will participate in some other manner, 3 in some other manner, and 2. So this total is 12 aldehyde molecules. These 2 aldehyde molecules, that is phosphoglyceraldehyde molecules, they undergo a change from aldehyde to ketone. The enzyme which helps is transketolase, and this transketolase would help in conversion of this phosphoglyceraldehyde 2 molecules into rubulose phosphate. So 2 molecules will participate in a different pathway. These other 5 molecules of phosphoglyceraldehyde, they undergo isomerization to form, again isomerization means this aldehyde will change into its isomer. So this isomer which is formed is dihydroxyacetone phosphate. These are the isomers. So here enzyme would be isomerase. These 5 molecules, here 5 phosphoglyceraldehyde are undergoing isomerization. So 5 of these will be formed. Now they divide again into 2 pathways. 2 follow one pathway and 3 would go through other pathway. The 2 which are here, the 2 dihydroacetone phosphate, they change into pseudohaptulose, and this pseudohaptulose changes into xylulose. This reaction which is taking place is again transketolation. So enzyme is trans-ketolase. And this xylulose which is formed, it gives rise to the rubulose phosphate. So ultimately out of 12, 2 followed a different pathway. With the help of trans-ketolase enzyme, they change into rubulose phosphate. 5 molecules, they follow another pathway. First they undergo isomerization to form a dihydroacetone. Then these 5, they again follow 2 pathways. 2 molecules go pseudoheptulose pathway, and 3 with the help of an enzyme aldolase will change into fructose 1,6-biphosphate. So they will form fructose 1,6-biphosphate. These 3 phosphoglyceraldehyde molecules, they straight away form fructose 1,6-diphosphate. So now how many molecules have changed into fructose 1,6-diphosphate? 3 phosphoglyceraldehyde molecules straight away through one pathway, and out of these 5, 3 again through aldolation. So 3 here from here and 3 from here. So if we keep a track of the carbon numbers here, these 3 molecules which are coming, they are 3 carbon compounds. So 9 carbons come from here, and same 3 molecules with 3 carbon, 9 come from here. So 9 and 9, 18. So 18 are here. Now 3 molecules are formed. 3 molecules of this. Fructose is a 6 carbon compound, so 6 carbon, 6 and 6, 3 molecules. Out of this, again there is a separation. 2 molecules follow a different pathway, and 1 goes to form glucose. So this 1 fructose forms. So when we say this phosphoglyceraldehyde separates to form glucose, it is not happening in one step. There are multiple intermediate steps which are taking place. So now out of these 3 fructose 1,6-diphosphate molecules, 1 separates to form glucose. 2 are still left. They form xylulose, again by the enzyme which helps is trans-ketolase. The enzyme is trans-ketolase. So now only 2 molecules are remaining, that is these last 2. These last 2 phosphoglyceraldehyde molecules, they form xylulose. So out of 12, all PGAL molecules do not follow the same pathway. 2 go another way, 5 undergo isomerization, and after isomerization again there are 2 pathways. 2 molecules follow the pseudoheptulose pathway, and 3 molecules will form fructose 1,6-biphosphate, out of which 1 fructose will give us glucose and 2 will come back into this regeneration of RUBP case. 3 straight away come to 1 fructose 1,6-phosphate and 2 form xylulose. So the difference is between the phosphoglyceraldehyde and the regeneration part. So these are all possible intermediate steps which actually take place. So when we talk of a simplified version, we straight away say that this phosphoglyceraldehyde is formed, 1 molecule or 2 molecules give us glucose and all others help in regeneration of RUBP. But Kelvin cycle, when it takes place, it is not a simplified version. Simplified version we take so that we understand the basic things first, and then when we understand basic, then addition of complex steps or intermediate steps becomes easier. So this is the actual Kelvin cycle which takes place in plants for carbon dioxide fixation. and this number which we have taken 12, we are assuming that everything is 6. So that is how we are getting 6 phosphoglyceric acids and 6 phosphoglyceraldehydes and everything, and that is how we have divided these intermediate steps accordingly. So now this completes our Kelvin cycle. We have understood the simplified version, which is good for our CBSE examination, and these intermediate steps are going to help us answer the questions of competitive exams. So after Kelvin cycle, the next thing which we need to understand is that do all plants follow the same pattern of photosynthesis? The answer is no. Reason is this Rubisco. In some conditions, when the temperature is very high, this behavior changes. The behavior of Rubisco changes. Instead of a carboxylase, it acts as an oxygenase. So if it starts behaving as an oxygenase, it's going to fix oxygen and release carbon dioxide, which is totally reverse of photosynthesis. So we will discuss that process plus the conditions which change the behavior of Rubisco.